All right, so let's continue by starting with lateral inhibition. And I'm going to use this picture from the slide to describe what it is. And then again, you can try to write this in your own words based on what I talk about. So lateral inhibition is a way to increase contrast of a physical stimulus in our somatosensory system. This also exists in the visual system, but in the visual system, lateral inhibition is to increase contrast of light and colors, not necessarily location. So in the somatosensory, which is what this question is asking about, because it says how does it identify location, which implies that we are talking about somatosensory here. In the somatosensory, it increases the contrast. So let's see how it does that. And this is directly from the lecture. So like you see in this picture to the left, you see that the pin is pushing down on this area of the skin and that the inner dark green circle is being highlighted because that is the most directly stimulated area of the skin from the stimulus. So we can see that the neuron that is most directly under that skin is the one that has the highest firing rate. Like you can see it's releasing more neurotransmitter here. And the secondary neuron will then also release a lot of neurotransmitter. However, we need our brain to know that this pin is touching exactly this point and not this point or this point over here. And the way we do that is with lateral inhibition. The fact that this axon will actually extend out to the peripheral neurons and inhibit them means that these neurons upstream, right closer to the CNS or in the CNS, will not fire at all. So when these neurons go to our brain, they just won't have a signal at all. There will not be a signal here going to our brain saying this area is being touched, which means that only signal B goes to the brain to tell our brain we are being touched in this specific place, which is this dark green spot. So that's how lateral inhibition works. It's the secondary neuron inhibiting the peripheral neurons so that the brain does not perceive the touch from coming from those places. Let's talk a little bit more about what is happening here with this inhibition. So like Dr. Chang talked about, this inhibition would most likely be the neurotransmitter GABA, and GABA would open ligand-gated chloride channels. So if chloride is entering the axon terminals of these peripheral neurons, well then they are going to make the cell more negative. So if that cell's not able to reach the threshold to open the voltage-gated calcium channels that is usually required for neurotransmitter release, then that means calcium won't enter, therefore neurotransmitters will not be released. So chloride is going to stop the release of neurotransmitters in those peripheral neurons. That might be a little bit more detailed than you actually need to know for lateral inhibition, but I just wanted to tell you that that's what is most likely happening in this scenario. But again, the overall function of lateral inhibition, increased contrast of location, or increased contrast of a stimulus location so that we know where that stimulus is on our body. All right, and I will quickly mention the visual system. The reason why this doesn't identify location in the visual system is because our visual system identifies location with if it is in our visual field or not. Like if we are able to see it, then we are able to identify its location. Lateral inhibition doesn't necessarily have anything to do with us seeing it in our visual field. Lateral inhibition does have something to do with the fact that we can distinguish contrast of colors in our visual field. So slightly different function, but it works the same way in the visual field. All right, the next thing I want to talk about is small versus large receptive fields and how this relates to rods and cones. All right, so over here on the left, and again, we'll kind of use a comparison between the visual and somatosensory systems to talk about these. So over here on the left, we have a high convergence. So we have high convergence, which relates to low resolution. Okay, so when we say a receptive field, we're talking about the neuron that is going to the brain. So we see that these secondary neurons down here are the ones that are actually going to relay this information to our central nervous system. It's not the primary neurons that do that, it's the secondary neurons. So because the secondary neuron is giving this information to the brain, then we say the receptive field of this secondary neuron is 1, 2, 3 neurons, so this entire space here that's circled. Let's compare this to this green secondary neuron on the right. So in this scenario, we have low convergence, which is related to high resolution. Okay, so again, if we are saying the receptive field is the one, or is related to the one that is going to send this information to our brain, then let's look at this secondary green neuron here. Well, this secondary green neuron is only getting its information from one other neuron, the green primary sensory neuron. So this green secondary neuron, its receptive field is only this small green area. So if you were to touch the purple area, your brain can actually distinguish that this is not the green area, that this is the purple area. And same thing, if you were to touch the green area, your brain can distinguish this is not the purple area, this is the green area. So the receptive field is referring to the neuron that is actually going to the brain. Now, the way this is related to our senses is the high convergence would be areas like your maybe arms or back or legs. So areas of your body that are small on the homunculus. And the small receptive fields would be the areas on the homunculus that are big, so hands and face. So your hands and face are big on the homunculus, which is the topographic map in the somatosensory cortex, saying we have more neurons dedicated to certain areas of our body than others, making our hands and face the most sensitive body parts, and areas like our back less sensitive. So we have a harder time pinpointing the exact location of a physical stimulus on our back than we do on our hand because of convergence here. Now, the way this is related to the visual system is over here, this would be rods, and over here, this would be cones. But more specifically, this is our central vision, which is the fovea. And over here, this is our peripheral vision. So, in our peripheral vision, we have very low resolution. The best example of that is you can't read with your peripheral vision, right? You can see things in your periphery, but you can't actually identify detail in it. Like if there's words in your peripheral vision, you could maybe identify that those words are there, but you wouldn't be actually able to read it. Whereas you can read with your central vision, your fovea. Where you're looking right now is the only place where you can read, because it is directly in your central vision, and that has the smallest receptive field so that you see with a very high resolution. That's why Dr. Chang was referring to like cameras when talking about this. So you actually only see with your central vision, at least you only see clearly with your central vision. You don't see very clearly with your peripheral vision, and that's okay, because our peripheral vision is really only meant to pick up motion and light and changes in light. So if we can identify motion in our periphery, then that's good enough. That's really all we need our peripheral vision for. But in our central vision, we need to be able to identify distinct colors, and we need to be able to identify things very clearly. So that's the difference between rods and cones in terms of receptive fields. So another part of this is, what is the advantage for each? Well, this is also kind of what Dr. Chang was talking about. The advantage of a large receptive field is that it is kind of cheap to make, in the sense of you don't need to dedicate a lot of sensory neurons to those areas. And that's kind of like the disadvantage for the small receptive fields, is you need tons of sensory neurons dedicated to those areas to make them very high resolution. So it's kind of more expensive from like a cellular level. It requires more cellular resources to have a small receptive field. All right, identify the differences between rods and cones. They're pretty similar, but they are for two different kinds of vision mainly. Like I said above, cones are for your central vision and rods are for your peripheral vision, but they are both photoreceptors. Cones are also for color, whereas rods are for kind of black and white, which is just basically light. And cones are in the fovea, whereas rods are in the periphery. But in the mechanism and how they actually do the phototransduction pathway, they're very similar. And that's what the next question is asking about. Explain the photochemical transduction in the rods and cones, and what is dark current? So let's talk about that. All right, so this is dark current. If you've studied for the MCAT before, this is the same thing as phototransduction. And like I said before, this is one of the moments where if you are maybe like a psych major and you've had to study sensory physiology before, you may have studied this in a lot of detail. In this class, we are still going to get into some detail, but the way I'm about to present it is the way that we are going to test it on. So I would suggest prioritizing this way, the way that we are presenting the material. All right, so this says it happens in rods. It does happen in cones. It is slightly different in terms of the proteins because the cones have the 3 different pigments. But for the purposes of this class, let's consider dark current, the mechanism that I'm about to talk about, to be the same in both rods and cones. They are very similar, but there are some little differences. But let's just consider them the same. Okay, so we have kind of 2 scenarios here. We have dark on the left and light on the right. So let's look at the absence of light in this first panel. So what we're looking at right now is like the inside of a rod. And a few key things to focus on here is whether the protein rhodopsin is active or inactive, whether cyclic GMP is high or low, and what the state of this channel is. So let's talk about what's happening here. So on the rod, you have 2 channels that can allow movement of ions. You have the CNG channel. CNG stands for cyclic nucleotide gated channel. So that's saying that this channel is gated, and the thing to open that gate is a cyclic nucleotide. Well, in this case, the cyclic nucleotide happens to be cyclic GMP. So if cyclic GMP is present, it will open this channel. Okay, I wanted to switch to black just to make things a little bit more clear. So if cyclic GMP is present, it will open the cyclic nucleotide gated channel. When that happens, calcium and sodium can move in because this channel is a non-selective cation channel, meaning calcium and sodium move in and the cell depolarizes. Now, the other thing that's happening on this membrane is we have potassium leak channels. So potassium will just naturally and slowly leak out of the cell, but when the CNG channels open, the influx of calcium and sodium is faster than the efflux of potassium, and the net result is depolarization. And again, this is a photoreceptor, so this is a special sensory receptor, so this is not an action potential, this is actually a receptor potential. All right, so let's look at what's happening down here. So it will depolarize and it will release a lot of neurotransmitter, and we know that this neurotransmitter is going to go to the bipolar cell, which will then go to the ganglion cell. So I'm just going to circle these two and consider these two as cells. Just by the way I've drawn it right now, just to simplify things. All right, so let's talk about like firing rates here. What would happen down the line? Now, when the photoreceptor releases this neurotransmitter, this says that the photoreceptor has a high firing rate. And in lecture, you learned that the function of the bipolar cell is to invert the signal. So if the photoreceptor has a high firing rate, the bipolar cell is going to have a low firing rate. And the bipolar cell will tell the ganglion cell what to do. So if the bipolar cell has a low firing rate, the ganglion cell will also have a low firing rate. So in the absence of light, we have no action potentials because the ganglion cell is not firing action potentials or has a low firing rate of action potentials. So the reason why the bipolar cell is able to invert the signal is because this neurotransmitter is actually inhibitory. And this neurotransmitter is actually glutamate, which in Unit 1 you learned that glutamate is a excitatory neurotransmitter, but this is an exception. In this case, it's actually inhibitory. Now, you don't need to necessarily worry that this is glutamate, but I would rather just focus on this is an inhibitory neurotransmitter. Now, if you read the textbook here, you will see that there are technically two different kinds of bipolar cells, where it's like an on bipolar or an off bipolar. Don't worry about that detail. Instead, focus on the fact that this neurotransmitter is inhibitory. It will make more sense conceptually. And that's what's more important, that you understand the concept of, in the absence of light, we do not have action potentials. We do not have action potentials. That's the concept here. So if you just think about glutamate here being inhibitory, if it inhibits the bipolar cell, it will reduce the firing rate of the bipolar cell, which will reduce the firing rate of the ganglion cell. All right, now let's use that concept of inhibition to see how do we turn light into an action potential. So in the presence of light, it will activate rhodopsin, and that process is called bleaching. And what happens here is when rhodopsin activates transducin, transducin is going to activate an enzyme called phosphodiesterase. And what phosphodiesterase will do is turn cyclic GMP into just GMP. So it's no longer the cyclic nucleotide, it is just the monophosphate nucleotide, which means that the cyclic nucleotide gated channel no longer has its ligand to open it. So with that phosphodiesterase getting rid of the cyclic GMP, this channel will close. And we see here that the calcium and sodium can now not enter the cell, or it will kind of bounce off like it's shown here. If it can't enter, then it'll just stay in the ECF. However, we still have potassium leak channels. So if potassium's slowly just going to gather out here, this cell will hyperpolarize. And when it hyperpolarizes, it will decrease the amount of neurotransmitter released. So right here, we say the firing rate of the photoreceptor has decreased. So again, if this is going to go to the bipolar cell, and if that is going to go to the ganglion cell, then let's talk about what happens here. If we have less inhibitory neurotransmitter, then we have more firing of the bipolar cell. So less inhibition will lead to more firing of the bipolar cell. And if the bipolar cell tells the ganglion cell what to do, then we will have more firing of the ganglion cell. So we see that here we turned light into an action potential here. So again, it's less inhibition leads to more firing of the bipolar cell. And this is the same concept that is used in the baroreflex to maintain your blood pressure. It's the concept of, do you have more inhibition or less inhibition? In the case of less inhibition, you get more firing. So we'll see this concept come up later. But this is the process of dark current or phototransduction. And again, like I said, we'll just consider this to be the same in both rods and cones, even if there is actually a very slight difference. All right, so you'll definitely need to go over this again because it's pretty complicated, but focus on these steps. Right, what is rhodopsin doing? Is it inactive or is it active? What is the concentration of cyclic GMP? And then 3, depolarize or hyperpolarize? So if you can kind of go through dark current in these 3 steps, is rhodopsin active or inactive? If it's active, it will decrease cyclic GMP. If it decreases that, it will hyperpolarize and cause an action potential. And then, and the opposite, if rhodopsin is inactive, it will increase cyclic GMP, which depolarizes, which decreases the action potential to your brain. So again, it's dark current because in the absence of light, you are actually generating the inflow of cations. You have a current, you have the movement of ions. So it kind of goes against what you would normally think a cell would do when it depolarizes and when it does not have the physical stimulus present. All right, but let's move on because we're almost done. And the last one is pretty easy. It says explain how the visual and auditory systems identify location. Like I said before, visual system is if it's in your visual field or not. That's how it identifies location. You have the right and left visual fields, and if it's in your vision or in your field, then you can identify its location. That's why Dr. Chang said it's not a problem for your visual system. You either see it or you don't. For the ear, it's also pretty straightforward. I will just mention that on the slide where he talked about this, he did go into a lot of detail, but he also put FYI for your information. You don't need to actually know all the angle differences in, like, if it's coming from like 45 degrees to your left, what is the time difference? You don't need to know that. All you need to know is the general concept that the way we identify location of an auditory stimulus is in the difference in time it takes to hit the left and right ear. For example, if there is a sound to the left of you, that sound will hit your left ear slightly before it hits your right ear, and when your brain processes this, it will perceive it as coming from the left. So that's how it identifies location. So this one is pretty straightforward. All right, that's all I have for this video. I had one more thing actually to mention with this picture. So I wanted to use this picture to summarize what's happening with dark current. So here, if we look at light coming in and striking the fovea, we know that light will hyperpolarize the cones and decrease their firing rate. And if they stop releasing the inhibitory neurotransmitter, they will increase the firing rate of the bipolar cell. And the bipolar cell will then increase the firing rate of the ganglion cell. And this is an action potential. So you turn light into an action potential. The other thing I wanted to point out with this picture is the convergence. So I'm going to just erase these just so we can see. All right, if you were to look at these cones here, the purple ones, for the most part, you see one cone go to one bipolar cell, right? This one goes to this one goes to this one goes to this one, and so on. But when you look at the rods up here, you see 1, 2, 3 rods go to one bipolar cell. So we would say that this bipolar cell has a large receptive field. Because of the high convergence. So because the rods are converging on the single bipolar cell, it's giving the bipolar cell a large receptive field, which means we have a lower resolution. So our peripheral vision, again, is for low resolution vision, it's really just there to detect motion, and it's mainly packed with rods, whereas our central vision, or fovea, is mainly packed with cones. So I just wanted to use this picture to point out what I had talked about earlier. All right, so I'll end the video here. I hope the study tips at the beginning of this video helped, and let me know if you have any questions in office hours.